Okay, let me continue now. The next chapter of second unit, and that is chapter number 6. Chapter 6, molecular basis of inheritance. Molecular basis of inheritance. Molecular basis. Now question is, what is this molecule that involved in the inheritance? Our earlier knowledge says that the main molecule involved in this inheritance is, and that is the DNA, deoxyribonucleic acid, and that is the molecule particularly play vital role in the inheritance. In the 20th century, many geneticists, evolutionists and naturalists and many other biochemistry scientists, they put forward so many theories regarding the inheritance character and that chemical component, and which is the chemical component, and that form the main molecule and that help in carrying inheritance character from one generation to another generation. And later it is come to know that is the one and only the molecule, and that is the DNA, deoxyribonucleic acid. And at present we know that DNA as the genetic material. DNA as the genetic material, and that also you are going to study in the next coming here the portion of the same chapter, that you have to deduce the DNA as a genetic material. So means here DNA is a genetic material, and the DNA only help in inheritance from one generation to the another generation. Our knowledge definitely says that here the inheritance character from one generation to another generation, and when Mendel explained what is that inheritance character, it was not properly understood, because at that time it was the beginning. But Mendel exactly known something that is passed from one generation to another generation, and that he called as a trait, or it is also called as a factor, or it is also called as in the general sense is a character. But what is this trait passed from one generation to another generation was not properly understood the Mendel and the Mendel time the scientist, because at that time the microscope, the electron microscope and other analytical biochemistry was not properly understood, and there is a lack of some knowledge also. But later on, in the middle of the 20th century, and then here number of geneticists, then biochemists come forward and rediscover the Mendel inheritance, and later it is come to know that the trait is nothing, the factor is nothing, and that factor nowadays we call as gene, and that study of gene is called as the genetics. That study of gene is called as genetics, and later on again it is discovered that what this gene contain, what is the chemical composition of the gene, and later it is again come to know that gene is nothing, gene is a molecule of DNA, deoxyribonucleic acid. So this is the main molecule that involved in the inheritance. About this you have to study in detail in this chapter. Now let us now start the DNA. About DNA you have to now start. So that what is the DNA? So DNA full form is deoxyribonucleic acid. Deoxyribonucleic acid, and this is the full form of the DNA. Now you know that DNA generally present in the nucleus, and that to be it is in the chromosome, a part of the chromosome. Understood? And later study again it is shown that a segment of the DNA is considered as a gene. Segment of the DNA is considered as a gene, and a small portion DNA, one small portion a gene. ಅಂದರೆ ಅರ್ಥ ಹೇಳಿ ದ ಜೀನಿನ ಕೆಮಿಕಲ್ ಕಂಪೋಸಿಷನ್ ಏನು ದ DNA. ಹಾಗಾಗಿ ವಿ ಹ್ಯಾವ್ ಟು ವಾಟ್ ಸ್ಟಡಿ ಹಿಯರ್ what first of all about the DNA, and then we can study more and more about the other things of this the molecular basis of the inheritance. Now first the DNA, deoxyribonucleic acid. Now this deoxyribonucleic acid here contain three main important chemical component, and that is first we will go to the chemistry of DNA. That is already you studied. Chemistry of DNA means chemical composition of DNA. DNA ದಲ್ಲಿ ಏನೇನು ಇರ್ತದೆ that already you studied in the first year science. Even then once again we will revise and see the DNA containing three main components. DNA containing three main components. That is one is nitrogen base, and the second one is pentose sugar, and then last one is phosphoric acid. Nitrogen base, pentose sugar, and the phosphoric acid are the three main components. See, nitrogen bases are further divided into two types. That is, one is furin and another one is pyrimidine. Furin and the pyrimidines are the two bases. Furin bases are again two types. That is, one is adenine A and another one is guanine. G. And then pyrimidines are two again. That is, one is cytosine and another one is thymine. And you also know that in RNA instead of thymine, what is present? Uracil. But you are not studying here RNA. You are studying about DNA. We will study later about the RNA also. But you must remember that only one changes in the RNA, only that pyrimidine base change, and everything remains same. Adenine same, guanine same, cytosine same, but only in RNA instead of thymine what is present, the uracil is present. That is only the change, and everything is the same. Everything is the same. So now these are the furin base. Then pentose sugar is the ribose, deoxyribose. Pentose sugar is what? Deoxyribose. And that may be the structure is like this, and it containing how many penta, and the 5 sugar molecule like this here. Pentose sugar, the formula C5H10O5. Adhe ನೀವು C6 ಇದನ್ನ hexo sugar ಬರೆದ್ರೆ ಅಂದ್ರೆ ಹೇಗೆ ಬರೀತೀರಾ? Hexo sugar is like this, no, and that is C6H12O6. This is hexose. ಯಾಕಂದ್ರೆ ಇಲ್ಲಿ 6 is 1 2 3 4 5 6, but here is only the 5 carbons are there. That is what C5, carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5, C5 and H10 and O5. That organic chemistry part you know, and here it is not required, and where again you study detail in the organic chemistry also. So again this is a pentose sugar C5H10O5, and then phosphoric acid is in the form of H3PO4, or generally it is phosphate. So these are the three main content or chemical composition of the DNA: nitrogen base, the pentose sugar, then phosphoric acid, and this. Then with this now we are going to next one, that is nucleoside and the nucleotides. What is this nucleoside and the nucleotide? In a DNA molecule or an RNA molecule, if nitrogen base plus pentose sugar present, and that is called as nucleoside. I think you understood. Andre bari nitrogen base pentose sugar idre ಏನಂತೆ nucleoside ಅಂತ ಕರೀತಾರೆ. If a DNA molecule is nucleoside plus phosphate present, and then it is called as nucleotide. Nucleotide ಅಂದ್ರೆ ಇದು ಮೂರು ಇದ್ರೆ nucleotide ಅಂತ ಕರೀತಾರೆ. ಮೂರು ಇದ್ರೆ nucleotide ಅಂತ ಕರೀತಾರೆ. ಅರ್ಥಾತ್ ಇದು ಮೂರು ಇದ್ರೆ nucleotide ಅಂತ ಕರೀತಾರೆ. ಎರಡು ಇದ್ರೆ ಮಾತ್ರ nucleoside. ಯಾವುದು ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಅಂದ್ರೆ pentose sugar and if all three are there, means nitrogen base, the pentose sugar, nitrogen base plus pentose sugar plus phosphate, and if all three are there, it is called a nucleotide. You remember, nucleotides are the structural and functional unit of DNA. Understood? Which are the monomers of the DNA? Name the monomers of the DNA. The monomers of the DNA is what? Nucleotide. The basic unit of DNA is nucleotide. Means, in other words, to form what DNA molecule and require what? Nucleotide. So means nucleotide get together form one long DNA segment. In other words, a long DNA segment that contains large number of nucleotide. Large number of what? Nucleotide. So in this way, here the nucleotide formation takes place. For example, one example we will take here. For the nucleoside and the nucleotide, one example we will take here. See, for what nucleoside, I will give you an example. Nucleoside. I will write first one. The first one is adenine. Adenine plus deoxyribose, pentose sugar, and that result in the form of deoxyadenosine. Deoxyadenosine is nucleoside. Deoxyadenosine is what? First nucleoside. Similarly, I write next one is guanine plus deoxyribose, deoxyguanosine, second nucleoside. Next one, cytosine plus deoxyribose, and it is deoxycytidine because it is a pyrimidine. You cannot write cytosine, cytidine, it is not a cytosine. Remember, because it is a pyrimidine base. These two are the furin base. If you are furin base, and next is the pyrimidine base, okay, cytidine, and then last one is it is a thiamine plus deoxyribose, and that is a deoxythiamidine. So these all are the what? Nucleosides. These all are the nucleosides. So all see how many nucleosides are there in the DNA. So name the nucleosides of DNA. In 2 marks question may be asked: deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythiamidine are the 4 important type of the what? Nucleosides. Similarly, you can also go for nucleotides. You can also go for nucleotide, and that nucleotide you can write here. See, nucleotide, which are the nucleotide? The nucleotide, now first one you want to write. The first, the nucleoside and phosphate. Nucleoside and phosphate result in the formation of nucleotide. That is why you have to write first one nucleoside deoxyadenosine plus phosphate or phosphoric acid, and that result in the formation of deoxyadenilic acid. Deoxyadenilic acid, or it may be also written as deoxyadenosine monophosphate, DAMP. Deoxyadenine monophosphate, ಇದು short form. ಸರಿಯಾಗಿ ಕೇಳ್ಕೊಳ್ಬೇಕು. There is no space to write there. Deoxyadenosine monophosphate, otherwise it is called. So name the nucleotide of the adenine ಅಂತ ಹೇಳಿದ್ರೆ deoxyadenilic acid. Second one, deoxyadenosine plus deoxyguanosine plus a phosphate, and that result in deoxyguanilic acid, and that also you can write DGMP, deoxyguanine monophosphate. Next, third one, deoxycytidine plus phosphate, and that result in deoxycytidine delic acid, and that can be written as DCMP, deoxycytosine monophosphate, and the last one is of deoxythiamine plus phosphate, and that will be written as deoxythiamidilic acid, or it is DTMP, deoxythiamine monophosphate. So like this, the four different type of nucleotides are there. You can write the nucleotides in short form also. You can write it in the short form also. DIMP, DGMP, DCMP, and DTMP. So these are the nucleosides and the nucleotides. Understood now? So in this way, here the nucleotides are the monomers of DNA, or they are structural and functional unit of the DNA. So this is about the the chemical component of the DNA. Then how these nucleotides are arranged in a DNA molecule? DNA molecule alli ee nucleotides alla hege arrange agtave adannu nodoಣ ಮೊದಲು. See, yes, as you know that, and here see, just I am giving you some structure, a DNA structure. Yes, this is a DNA structure, and here see, here what happens is, in a DNA, double stranded it is. Double stranded DNA it is. The double strands are there. This is a one strand and this is a one strand. This is a 5 prime, this is 5 prime, and this is 3 prime, and this is 3 prime, and this is a 5 prime. And that depending upon what here, the sugar phosphate molecule, the attaching position, okay, 5 prime and the 3 prime, and then here is also 3 prime and the 5 prime ಅಂತ ಕರೀತಾರೆ. So and that is a region where hydroxyl group and the phosphate group attach. ಅದಕ್ಕಾಗಿ ಅದಕ್ಕೆ 5 prime and 3 prime ಅಂತ ಕರೀತಾರೆ. Now these are the two strands just like a ladder. ನೋಡಿ, ಈ ಎರಡು ಸ್ಟ್ರಾಂಡ್ ನಮಗೆ ಲ್ಯಾಡರ್ ಟೈಪ್, ಒಂದು ನಾವು ಸೀಟಿನ ಅಥವಾ ಏಣಿ ಅಂತ ಏನು ಹೇಳ್ತೀವಲ್ಲ, ಆ ಏಣಿಗೆ ಎರಡು ಕಂಬ ಬೇಕು. ಆ ಎರಡು ಕಂಬದ ಮಧ್ಯದಲ್ಲಿ ಏನನ್ನ ಜಾಯಿಂಟ್ ಮಾಡ್ತೇವೆ ನಾವು, ಸ್ಟೆಪ್ಸ್ ಅನ್ನು, ಅಂದರೆ ಇಲ್ಲಿ ರಾಡ್ಸ್ ಅನ್ನು ಹಾಕ್ತೇವೆ. ಆವಾಗ ಇದು ನಮಗೆ ಏಣಿ ಆಯ್ತು, ladder. That is what we call as a ladder. Similarly, here in the DNA molecule, there is a ladder-like structure, but it is a helically coiled. Ladder-like structure, but it is what? Helically coiled. ಅದು helically coiled ಆಗ್ತದೆ. ಈ helically coiled ಅನ್ನುವಂಥ ಸ್ಟ್ರಕ್ಚರ್ ಈ ಟೈಪ್ ಕಾಣಿಸ್ತದೆ, ರೈಟ್ ಹ್ಯಾಂಡೆಡ್ ಫ್ಯಾಷನ್ ಅಂತ ಕರೆದಿದ್ದೀವಿ. ಸೊ ಹಾಗೆ ಹೌ ದೀಸ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅಟ್ಯಾಚ್ ಅಂತ ಹೇಳಿದ್ರೆ, ಒಂದಕ್ಕೊಂದು ಅಟ್ಯಾಚ್ ಆಗಿದ್ರೆ, ಸಿ, ಆ್ಯಂಡ್ ಹಿಯರ್ A double bond T, G triple bond C, T double bond A, C triple bond G, C triple bond G, G triple bond C, A double bond T, like this, in this fashion. In this fashion only it undergoing for the pairing. Here adenine always combine with the thymine with the 2 hydrogen bond. Adenine always combine with the thymine with the 2 hydrogen bond, or vice versa. Thymine always combine with the adenine with the 2 hydrogen bond. So you must remember that adenine is a furin base and thymine is a pyrimidine base. It always one furin base and the one thymine pair, and that to be it is restricted. What restricted? Adenine is a furin. No, adenine is a furin. It always combine with the pyrimidine of thymine, not with the cytosine. That you must know. Understood? See, adenine double bond thymine. Adenine never combine with what? Cytosine. Idu agodilla. Not will happen. Since cytosine is also pyrimidine. In other words, adenine always pair with the thymine, or vice versa. Similarly, guanine always pair. Guanine is what? The furin base. Always pair with the cytosine with a 3 hydrogen bond, or cytosine always combine with the guanine with a 3 hydrogen bond, vice versa. But this is also restricted. In other words, the G never combine with the T, or G not combine with the A, since they are same furin, it will not happen like this. So that is why you must remember that in an anti-parallel DNA, here pairing is taken place, and at that time adenine always combine with the thymine and guanine always combine with the cytosine, or vice versa it may be. But adenine with the thymine 2 hydrogen bond, and guanine with the cytosine with 3 hydrogen bond. This also you have to remember, and this is also restricted. So like this, here the DNA double stranded system taken place, and here the single polynucleotide chain you have to study. One single polynucleotide chain book I have already given the notes also, and that notes also you can refer. Single polynucleotide chain, one single polynucleotide chain one single polynucleotide chain, and that is to be in C. How it is ಅಂತ ಹೇಳಿದ್ರೆ, and this is a single polynucleotide chain ಹೇಗಿರ್ತದೆ, C H H, and then it is a P, and in the other side it has a connection with the what? Connection with pentose sugar. And this pentose sugar in other side is connected with the nitrogen base like adenine. Like this connection is the E type of connection irthude. And then similarly in the next again there is another phosphate will be there here, phosphate, and then C H, and then again there is one more pentose sugar. ಇಲ್ಲಿ ಮತ್ತೊಂದು pentose sugar ಬರ್ತದೆ, and that may be connected with the maybe a guanine like this, got it? And then once again here the phosphate, and then again H. You can go to the book also, and here is again the one more pentose sugar here like this, and it may be again the T pyrimidine base like this. So this is one polynucleotide chain, means one side of the matra. So like this here, see this is a backbone. This is a backbone. This is lateral. This is a backbone. Sugar phosphate. Here is also sugar phosphate. So means this the ladder pillar. This ladder pillar, and this pillar is made by. This pillar is sugar and phosphate. Here is also sugar phosphate, and this is connected by adenine double bond thymine. Then next one is guanine triple bond cytosine. Next one cytosine triple bond guanine, and then adenine double bond T. Then thymine double bond A. ಅಂದ್ರೆ ಈ ಎರಡು ಏನು ಪಿಲ್ಲರ್ ಅಂದ್ರೆ ಬ್ಯಾಕ್ ಬೋನ್, ಶುಗರ್ ಫಾಸ್ಫೇಟ್ ಬ್ಯಾಕ್ ಬೋನ್ ನ ಕನೆಕ್ಟ್ ಮಾಡಲಿಕ್ಕೆ ಇಲ್ಲಿ ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಅಂತ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ that help, and that only help in the the connection of this, and that result in anti-parallel strand. And exactly anti-parallel strand ಎಲ್ಲಿಗೆ ಟರ್ನ್ ಆಗ್ತದೆ, ಹೆಲಿಕಲಿ ಕಾಯಿಲ್ ಆಗ್ತದೆ ಹೇಳಿದ್ರೆ ಇದರ ಮಧ್ಯದಲ್ಲಿ 1 2 3 4 5 6 7 8 9 10 ಎಲ್ಲದು 10 ಇಂದದು ಬಂದ್ಬಿಟ್ಟು ಅದು ಕಾಯಿಲ್ ಆಗ್ತದೆ. And that's length is about 3.4 angstrom minute. Its length ಏನು 3.4 angstrom minute, ಅದು 54 nm ಅಂತ ಬರ್ತದೆ. 3.4 ಅಂದ್ರೆ ಇಲ್ಲಿ ಕಂಪ್ಲೀಟ್ ಇಲ್ಲಿಂದ ಇಲ್ಲಿವರೆಗೆ ಡಿಸ್ಟೆನ್ಸ್, ಇಲ್ಲಿಂದ ಈ ಪಾಯಿಂಟ್ ಇಂದ ಈ ಪಾಯಿಂಟ್ ವರೆಗೆ ಡಿಸ್ಟೆನ್ಸ್ ಏನಿದೆಯಲ್ಲ ಅದು ಎಷ್ಟು 3.4 angstrom minute, ಸಾರಿ 34 angstrom minute ಆಗ್ತದೆ. ಅದು 34 angstrom minute, and the each distance of this is what 3.4 angstrom minute ಆಗ್ತದೆ. ಸೊ ಅಂದ್ರೆ ಒಂದು ಇದರ ಡಿಸ್ಟೆನ್ಸ್ 3.4 ಆಗ್ತದೆ, and 34 angstrom minute. So like this here distance between each is also the same, ಅಂದ್ರೆ iso distance of. So one same distance ಇರ್ತದೆ. So that is why it is very important the aspect. It may be asked in the 1 marks question also. So remember this, it is also very important. So one minute. Yes. So in this way, the anti-parallel strand helically coiled and arrangement of adenine, the thymine, guanine, cytosine, etc. also taken place. And this is a polynucleotide chain. This polynucleotide chain is next double stranded bರ್ತದೆ. That also you can go to your book. You know the book ಅಲ್ಲಿ ಬಂದ್ರೆ ಈಗ adenine combined with thymine. ಈಗ thymine ಗೆ ಇಲ್ಲಿ ಮತ್ತೊಂದು the pentose sugar ಇರ್ತದೆ, and this is a pentose sugar and this is pentose sugar, and to this pentose sugar and there is what the C, H and then P. Correct? ಇದಕ್ಕೆ T connection ಆಯಿತು. And then once again to this one is one more what connection ಬರಬೇಕು ಈಗ C, H and then here one more pentose sugar connection. Pentose sugar connection attached to this. Sorry, triple bond C, and this is connected. So means A double bond T or T double bond A, C triple bond G or G triple bond. Andre ಈಗ double stranded ಆಯ್ತು. This is a one strand polynucleotide chain and this is another polynucleotide chain. These two polynucleotide chains are connected by means of hydrogen bond. Exactly what shown here the same. Exactly what is shown here is the same. So in this way here what these nucleotide get with one another by pairing with neighboring this strand, and thereby it get connection and the result in the formation of double stranded. Now the nucleotide nodiga, say this is a nucleotide. So nucleotide is in the sense here. So this is a nucleotide. Nucleotide andre ಈಗ nitrogen base pentose sugar phosphate. See, nitrogen base pentose sugar phosphate. This is one nucleotide. So this is one nucleotide, and this is the nucleotide. This is one nucleotide, correct? This is one nucleotide, and here is also another nucleotide. So means this is nucleotide and this is nucleotide, correct? And the nitrogen base pentose sugar phosphate, nitrogen base pentose sugar phosphate, and this nucleotide and this nucleotide what pair with the hydrogen bond, and that result in the the pairing. So means that each nucleotide of the DNA that is a monomer and are the structural and functional unit of the DNA. So here is also happen same time. So this is a nucleotide, this is one nucleotide, and here is also the another nucleotide, and that should be here the phosphate. And so like this means it is not continued here, and here should be the phosphate, and if it is so, is also nucleotide formation taken place. So in this way here, see sugar, pentose sugar, the nitrogen base, all these are the nucleotide, and all nucleotide get together to form one long chain on the DNA strand. In another side also there is a long chain of DNA strand formation taken place, and these are what are paired with the hydrogen bond, and that is the restricted nitrogen bases. So this is about the DNA and the DNA structure. The characteristic feature also you studied in the online classes during the Zoom application, and that also you can just read by your notes, and next class we will continue the remaining of the this molecular basis of inheritance. Okay.